Hello everyone, welcome to our next lecture on basic cell structure and function. So in this lecture we're going to talk about membranous organelles, we're going to talk about the nucleus, the endoplasmic reticulum, the Golgi apparatus, mitochondria, and then vesicles, lysosomes, and peroxisomes. So we'll talk about the nucleus first. So the nucleus is going to be the largest organelle in the cell. It's the only one that's visible, really, with a light microscope, and the nucleus controls cell function. So the first structure that's part of the nucleus is called the nuclear envelope. This is going to be basically the membrane that surrounds the nucleus. Now, remember, membranes are made of phospholipid bilayers. The nuclear envelope is the first example of an organelle that actually has two lipid bilayers. So that's why this is called an envelope, right? Not the nuclear membrane, because membrane would sort of indicate that there's only one lipid bilayer, but here there's actually two lipid bilayers and then a little space between them. So it's called the nuclear envelope. Kind of picture an envelope, right? It has two walls and a space in the middle. Now, inside the nuclear envelope, you can see these little orange structures. These are the nuclear pores. These are selective pores that allow things to move into and out of the nucleus that are made of membrane proteins. And by selective, this is important because you don't want just anything to get into and out of the nucleus. The nuclear pores can kind of, pick and choose what enters and what doesn't. And so, they're really important in that respect. Now, inside the nuclear envelope, you're going to have a liquid that sort of fills the nucleus called nucleoplasm. And inside this nucleoplasm, you're going to have a nucleolus, which you typically see also with a light microscope as a sort of a dark spot inside the nucleus. That nucleolus produces components that make up ribosomes and a form of RNA called ribosomal RNA or rRNA. And then throughout the nucleoplasm, you're going to have this substance called chromatin. I'm going to switch slides for that. Okay. So chromatin is going to be DNA and protein. So inside the nucleus, the DNA strands, which you can see here, are going to be wrapped around these proteins called histones. Now, the reason you have to do that is because the DNA strands inside your cells are actually really, really, really long. If you were to take all the DNA out of the nucleus of a single cell and you were to line the strands up end to end, they would stretch to be about 2 meters long, right? 2 meter sticks long. That is incredibly long, and you're going to have to take something like that and wind it up around these histone proteins really tightly so that it fits into this nucleus that you can barely see with a light microscope. And so chromatin is really the DNA wound around these proteins. Okay? Now, DNA in the nucleus is going to be found in 46 separate strands called chromosomes. All right, so sorry guys, this didn't have any voiceover before, so I'm redoing it. But I mentioned to you guys about chromosomes in the previous slide. So this is where I talk a little bit more detail about these chromosomes. So we have to talk about the two cell types, what are called somatic cells and gametes. So somatic cells are most cells in the body, basically any cell in the body that's not a gamete. Gametes are sperm and egg only and nothing else. Okay? Now, the reason we have to talk about these as different is because they're going to have different numbers of chromosomes. Sorry about that. Somatic cells are going to be what are called diploid. So diploid means that they're going to have two copies of each chromosome. You're going to get one copy of each chromosome from your mom and one copy of each chromosome from your dad. So that means 23 from your mom, 23 from your dad. So that means in diploid cells like your somatic cells, you should have 46 total chromosomes. Now the image that you're seeing here is called a karyotype. And a karyotype, you take a cell in the middle of mitosis, you kill it, you pop open the nucleus, and you stain the chromosomes. Now you do this in the middle of mitosis because this is when the chromosomes are they're most dense and actually you can see them with a light microscope. So you stain them so that you can see the banding patterns, and you use the banding patterns and the size and the position of the little centromere to match the chromosomes to their pair. So you match the maternal to the paternal for chromosome number 1, and the maternal to the paternal for chromosome number 2. So you can actually see that, from this cell, I have 2 copies of every chromosome, 1 to 23, so that means 46 total. Now, chromosome pairs 1 to 22, these are what are called autosomes, and they're just chromosomes that determine basically everything except genetic sex. Sex chromosomes are going to be just the last pair, the 23rd pair of chromosomes. Okay? Now, most of these chromosomes, with the exception of those sex chromosomes, particularly in males, are going to be what are called homologous. So homologous means similar but not exactly the same. So that means they carry the same kind of information, but not, sometimes you get different information from your parents, right? So the example I gave is this gene for PTC tasting. So PTC is like this chemical where they put it on a piece of paper, and some people taste the paper, it tastes bitter, but other people taste the paper and they don't taste anything. And it's based on this gene called the TAS2R38 gene on chromosome number 7. So you can inherit the gene that allows you to taste it, or you can inherit the gene that doesn't allow you to taste it. Right? In both cases, the gene for PTC tasting, but one gene is for tasting and the other is for not tasting. So you can see how, like, on the same chromosome, you can have the same genes, right? The same information for, say, PTC tasting, but that information can be different in different parents. Sometimes it could be the same. Maybe sometimes you get, the tasting gene from both your parents, but other times you can get something slightly different where you get a tasting gene from one parent and a non-tasting gene from the other. And in that case, it's slightly different. So that's what we have to say homologous, basically meaning similar but not the same. So that's what you should think, that these chromosomes carry the same kind of information, but not always exactly identical. Right? Now, with the sex chromosomes, one of the things that's really going to be very different about them is that there are actually 2 types of chromosomes. There's an X chromosome and a Y chromosome. And they're actually not the same size, and they don't carry the same kind of information. So what you're seeing, sorry, let me jump back to the laser pointer. What you're seeing in this karyotype here is the karyotype from a female. So if you're female, you get an X chromosome from your mom and an X chromosome from your dad. So you have 2 X chromosomes as your 23rd pair of chromosomes. Okay? So in this case, if you're female, your sex chromosomes are really similar to every other kind, right? The 2 chromosomes are homologous. They're the same size, they carry the same amount of information and all that. However, the other sex chromosome is going to be the Y chromosome. And if you were to see the Y chromosome, I'm going to switch to a pen here. You see the Y chromosome. The Y chromosome is about a third the size of the X chromosome. And it carries very, very little information. The only thing that the Y chromosome carries are the genes that produce male genitalia, and they carry essentially nothing else. So if you're male, you get an X from your mom and a Y from your dad. But basically what that means is that all the genes on the sex chromosomes on this 23rd pair, if you're male, you essentially inherit all of them from your mother because your dad just gives you the genes for male genitalia and nothing else. So the sex chromosomes are going to be a little bit different than all the other types. So next is the endoplasmic reticulum. So this is often just abbreviated as ER. So the endoplasmic reticulum is going to be a network of membranous envelopes or pouches that are called cisternae. So you can see those as the blue structures here. Okay? So those are the membranous pouches that are called cisternae. Okay? So these cisternae are spaces, are sites where the cell synthesizes molecules, basically where it makes molecules. And in general, whenever you think of the endoplasmic reticulum, that's what you should think. This is a region of the cell that makes things. Okay? Now, endoplasmic reticulum comes in two types. What's called rough endoplasmic reticulum, or rough ER, or sometimes just abbreviated as RER, and smooth endoplasmic reticulum, or what's abbreviated as smooth ER, or sometimes just SER. Okay? I usually say rough ER and smooth ER is usually how I say it. Okay? So rough endoplasmic reticulum has ribosomes attached to it. And so you see that one as the blue cisterns with the orange little spots. Those are the ribosomes attached to the surface of the rough ER. So it's called rough like this because when you look at it under the electron microscope, it looks like there's a bunch of spots on the surface. And I'll show you guys that in another image later. Now, rough endoplasmic reticulum is going to produce membrane proteins, and it produces secretory proteins. So it produces the proteins that are going to be in the membrane. Remember the transmembrane proteins that we talked about? Those are produced here. And then also secretory proteins, those are proteins that will ultimately be transported out of the cell, basically to do something outside the cell. And you can sort of see that going on right here. Okay? Smooth endoplasmic reticulum, or smooth ER, is going to be cisterns that don't have ribosomes attached to the surface of them. So notice this here. Okay? Notice that that's the blue cisterns. They're skinny, but they're still cisterns, and they don't have any of the orange dots on them. Okay? Now, because they don't have these ribosomes, that means the smooth ER isn't going to make protein. You guys will learn later, we do the non-membranous organelles. Ribosomes produce proteins. That's one of the things they do. So that's why rough ER produces proteins, but smooth ER doesn't because they don't have any ribosomes. That means no protein production is going on here. Okay? So then, since smooth ER doesn't produce protein, what does it produce? Okay, it produces the other kinds of macromolecules in the cell. Smooth ER produces the carbohydrates, it produces the lipids that the cell will use to perform whatever tasks it needs to perform. The other thing that smooth ER does is it actually stores calcium for cells. And that'll be important in all cells, but particularly important in muscle cells. So this is the image I told you. This is an electron microscope image. And the pouches that you're seeing here and then also here, those are the cisterns of the ER. And then notice that the rough ER has these dark spots on the surface, but when you look at the smooth ER, you don't see any of these dark spots. Okay?